I'm going to go a little bit more into depth about proteins and nucleic acids in this one. So as a reminder, proteins have a central carbon, they have an amine group, a carboxylic acid group, an R group, which in this case is represented as CH2SH, and an H off the central carbon as well. As a reminder, when we're growing a chain of amino acids to make a protein, we're always adding to the carboxylic acid side. This amino acid is one of the 20, and it is cysteine. Sometimes we abbreviate amino acids by 3 letters. Sometimes we show an amino acid as a circle instead of drawing out all of the elements. As a reminder, R groups can be hydrophilic, hydrophobic, basic, or acidic. We use those models in class where different colors represented the different properties of the R groups. And you'll remember that when you folded those models, the hydrophobic R groups ended up on the inside. They were yellow in the kits. Hydrophilic blue groups ended up on the outside, and acidic and basic groups, because they're positive and negative, would be attracted to each other. All of these things end up leading to the structure of the protein as it's folding. There are 4 levels of protein structure. Primary is the order of the amino acids. So the order matters. If we switched arginine or valine or put a different amino acid in there, it would change the way that the protein ends up folding. So the primary structure is very important. Secondary is the attraction between the carboxylic acid group of one amino acid and the amine group of another amino acid to make these patterns called alpha helices and beta pleated sheets. The bonds between those carboxylic acid and nitrogen groups are hydrogen bonds that are kind of lightly holding the alpha helix and beta pleated sheet in place. Tertiary structure is what I was showing with this picture. This is when we have the R groups interacting with each other to form the 3-dimensional structure of the protein. This includes hydrogen bonds between partial positive and partial negative areas, ionic bonds between fully positive and fully negative areas, covalent bonds. If that's happening between R groups, that's always between sulfides, always between cysteine, and we call that a disulfide bridge. Also, hydrophobic and hydrophilic interactions help to shape the molecule as well, as we saw here where the hydrophobic groups are kind of clumped in the middle. Quaternary structure is when multiple strands of protein interact with each other. So like if this green protein that had folded up on its own and this purple protein that had folded up on its own are now interacting with each other. And the same types of bonds apply. We're just between separate proteins at this point. So there's still hydrogen bonds, ionic bonds, covalent bonds that are holding those in place, but it's between different proteins. And often a functional enzyme or a functional protein will have multiple individual primary chains in it. So for example, hemoglobin has 4 protein strands that interact with each other to be its functional hemoglobin. Nucleic acids, as a reminder, a more detailed view of what the monomer looks like. We have a phosphorus group, phosphate group. We have a sugar. The sugar is different in DNA and RNA. And we have the nitrogenous base, and that's different depending on the letter A, T, C, G, U, etc. In DNA, it's double-stranded. We have a 5' end and a 3' end. Remember, 3', 3 is free. That's without the phosphate group. Also remember that the DNA strands run anti-parallel, meaning if this one's 5 to 3, the opposite would be 3 to 5. Notice how it looks like one is upside down from the other. And there's hydrogen bonds that hold the nitrogenous bases together. A pairs with T, G pairs with C. Quick reminder, G and C have 3 hydrogen bonds between them. A and T have 2 hydrogen bonds between them. This is what breaks when DNA is opening to be replicated or be transcribed. When we're adding another nucleotide onto a growing strand of DNA, we're always adding to the 3' end. The 3 is free end. The way I remember that is the DNTPs, or the nucleotides that are about to be added, have 3 phosphates just like ATP, and that's where the energy comes from to make that bond between this nucleotide and the strand that's already there. So we always add to the 3' end. Some reminders about DNA versus RNA. DNA is double-stranded. The sugar is deoxyribose. It has T. They both have ACG, whereas RNA has U, uracil, instead of T. RNA is single-stranded, and the sugar that it has is called ribose.